IA, PTDP for short, covers hit to lead characterization, IND enabling studies, and phase 1 clinical trials. And then there are several other programs from NCATS and the preclinical screening program for pain that cover translational assays and preclinical therapeutic profiling. I'll just tell you a little bit about PSPP in a minute. And we also cover a phase 2 clinical trial through the EPPIC-Net program. And you can see on the slide that there are quite a few participating NIH institutes and offices so that we can cover pretty much all types of pain with these grants. Now, for a little information about the preclinical screening program for pain, or PSPP, this program is set up to provide a platform to identify and profile non-addictive, non-opioid therapeutics. The program accepts biologics, small molecules, natural products, and devices, and it moves them through a series of in vivo pain models and pain testing in animals, so that the idea is that assets that move through these could possibly go towards clinical trials, or at least the information could provide an important addition to the package that might be used for investors. Under NINDS direction, the PSPP program provides rigorous preclinical evaluation of assets in a suite of assays that are performed by contract facilities, and Psychogenics is our contractor. And this is all done in a blinded and fashion and a confidential basis at no cost to the participant. The PSPP staff interacts with the participant to plan testing, review test results, and discuss future studies. The participant intellectual property and confidentiality are protected and are very important considerations for us. PSPP is open to academic, industry, and government asset owners worldwide. And if you want to get more information about this really interesting and excellent program, please contact the program director, who is Smriti Iyengar, and this is her contact information on the slide. And as Rakanda said, these slides will be available later after this is over. Now I just want to move to the actual Heal Pain Therapeutics Development Program. That's the topic for today. This program is a unique, I think, provides a unique virtual pharma approach that features industry experienced consultants and NIH contract resources. It spans hit to lead optimization all the way through phase 1 clinical trials. It supports biologic and small molecule therapeutic developments. Another feature of it is that NIH consultants are assigned and tailored to each project that is awarded based on the needed expertise for the project. NIH contract resources are tailored for each stage of the project too. And so in addition to consultants, we also offer CRO or contract resources that cover various functional areas involved in drug discovery and development, which you can see on the right-hand side of the slide. A grant awardee can choose which NIH contracts to use, or they can opt to budget all their own contracts in the grant proposal. Often our grantees choose to do some of the work on their own and then use some of our contracts. And then most importantly, the investigator team's intellectual property always will be retained by the investigator's institution. We seek to have all IP go to the PI. And the diagram shows you a little bit about what a lead development team looks like. Once an award is granted to an applicant, a lead development team is formed around that project. And the team consists of the principal investigator in the project or the grantee, and also industry seasoned consultants who have expertise in assay development, medchem, CMC, pharmacokinetics, and drug metabolism, or pharmaceutical development. And then NIH staff are also on the team and are the project managers who coordinate the team. And then you can see from the diagram the different resources that are provided in addition to the consultants and the NIH staff. So the NIH grant itself, of course, provides support for the entire project. And the only thing about that is that although we provide contracts for most cross-functional areas involved in drug discovery, it is up to the investigator to provide bioactivity and efficacy study work on their own, and so they can budget that into their grants. NIH contracts where we provide the support are involved in medicinal chemistry, and Curia is our vendor for that. PK and toxicology, SRI, Toxicology Research Labs, ITRI and TRL are all vendors for the PK tox contracts. Data management contracts are provided by the Collaborative Drug Discovery, CDD, and manufacturing and formulation. We have contracts from Curia, MRI Global, ADL, Battelle, and Leidos to support those types of activities. And then with clinical trials, Duke University and PPD are our vendors for those. Then I should point out too that we, for 2024, the receipt dates, and these are the same ones that are for resubmission dates, are January 24th, May 24th, and September 24th. In this slide, we thought it'd be nice to just point out that, we support a full spectrum of different types of targets. We really just review looks at the scientific quality of the proposals, not necessarily the targets or anything, but we're happy to say that we have a really good variety of targets. And we have biologic targets, small molecule targets. We even have some approaches involving formulation changes and combination device and drug combination. So we do offer a fairly large diversity in approaches towards pain therapeutics. So some of the features of a grant that you would get from the Pain Therapeutics Development Program involve the following. So some of these features are that the grant itself might be a little different if you're used to R01 grants. This is a different one in that it's a phased grant. It has two phases, a UG3 and UH3 phase, and it's a cooperative agreement, which means that the NIH has a substantial role in working with you if you're awarded one of these grants. The other important thing to realize is that these grants are milestone driven, so that in your application, you want to propose milestones for each year. And those metrics determine whether or not you proceed to the following year of funding. Another thing the grant features are that it supports the early therapeutic development process, not just one part of the process, but the entire process, which includes, so I was mentioning, hit to lead activities, lead optimization, selection, characterization, biomarker optimization, and PK/PD development, and IND enabling studies and phase 1 trials. The end goals for this program are to identify and fully characterize a lead candidate. Another one is to identify target engagement biomarkers if you can. A third one is to seek partnerships. Our main goal is to commercialize your therapeutics that you successfully discover and develop. And one important thing is to get beyond phase 1 if you make it that far. So it's important to seek partnerships while you're funded with these grants, and to complete IND enabling studies, file the IND, complete phase 1, and then in that sense, then get ready for phase 2. We don't support phase 2 or some of the readiness activities for phase 2, but we try to get the grantees to be thinking about that and planning, especially as they approach the last several years of the grant. So one thing about these grants is that they're fairly flexible in that applications can enter in either a discovery or development stage. But one thing that's important to remember is that, remember I said it was a 2-phase grant, the UG3 and the UH3. So all projects, whether they enter in discovery or development, must start with the preparatory UG3 phase, and then they end with an implementation or execution UH3 phase. In the entry phase, for discovery, you might imagine some of the entry criteria are different than if you're in the development stage. So for the discovery stage, you must show that you've completed the entry criteria for hit selection. For the development stage, you must show that you completed the entry criteria for IND enabling. And those are all specified in the funding opportunity announcement. The duration of the UG3, UH3 combined is 5 years. And just as a general guideline, usually the UG3 phase is for 2 years, and 2 years is the maximum number of years that you can apply for the UG3 phase. And then for a total of 5 years, the UH3 phase then is the remainder of the grant, whether it be 3 years if you chose to have UG3 for 2 years, or if you chose to have UG3 for 1 year, then of course it'd be 4 years for UH3. And again, we can't emphasize enough, in your application, you must include plans for both the UG3 and UH3 activities in the description of your approach and in your budgets. And then another important thing to remember is all projects are expected to lay out a path for the clinic through the grant or NIH contracts. And this is just to show us that you've taken into account all of the work process involved to go from discovery through phase 1, or if you're in the case of development, from the IND enabling through phase 1. Then a little bit about eligible organizations. So academic institutions are eligible, small businesses are eligible, and organizations with foreign components that are proposing to use foreign components in subcontracts or as subawards are also okay. One thing that's different with this is the this will be the first receipt date in January for our reissued version of this funding opportunity. The reissued version does not allow a foreign institution to be the main grantee. We still allow foreign components, but as I mentioned, if you're a foreign institution and you want the grant support to go through your institution, then that is not going to work. So just keep that in mind. And examples of, you might wonder what a foreign component is. Some examples of grant-related activities that would be using foreign components are collaborations with investigators at a foreign site anticipated to result in co-authorship, use of facilities or instrumentation at a foreign site, or receipt of financial support or resources from a foreign entity. Foreign travel for consultation is not considered a foreign component. Just want to talk a little bit now about entry criteria for people who are entering the program in the discovery stage or the development stage. If you're entering in the discovery stage, you need to provide a rigorous biological rationale for the intended approach. You must have a promising small molecule or biologic starting point for optimization. And you must show that you have scientifically sound assays to optimize and test the agent. If you're entering in the development stage, the candidate therapeutic should already be identified. You should provide a strong data package that links the modulation of the therapeutic target by the candidate therapeutic to disease modification. You should also show biological activity and admit properties of the candidate therapeutic that are appropriate for the intended use. And here, just a little bit about the 2 phases. So the preparatory or UG3 phase supports things like optimization using potency and efficacy screens. And that, of course, would be in the early discovery stages. It also supports things like obtaining preliminary efficacy and toxicology testing in appropriate animal models for pain. That would be important, even if you're entering the discovery stage or the development stage. Characterization and testing for in vitro ADME. And of course, just to remind people, ADME stands for absorption, distribution, metabolism, and excretion. And again, those things are done both in discovery and development stages. And then initial optimization of pharmacodynamic target engagement biomarkers that are associated with the target or pathway. And this is an activity that's also supported in discovery and development phases of entry. For the execution phase, activities are things like any further optimization activities as listed in the funding opportunity announcement. Things like pharmacokinetics, biodistribution, gene expression, tumorigenicity, and immunogenicity are things that would be looked at in the UH3 phase. Cell bank development and testing, same way. Formulation and manufacturing for toxicology and human clinical studies, again, in the UH3 phase. And IND enabling characterization of the clinical candidate, of course, in the UH3 phase. Non-GLP and GLP tox studies, and IND submission and phase 1 clinical testing, all in the UH3. So want to point out a couple other things about applying. So there are some reasons where your application could be withdrawn by the scientific review and the receiving referral groups at NIH. And it would be for things, one reason for application withdrawal is non-responsive activities. And some of these things that are non-responsive are screened for initial hit identification, basic research and studies of disease mechanism, animal model development, development of diagnostics and diagnostic devices, clinical studies beyond phase 1 testing. And this is important, this last one, opioid sparing projects and projects targeting the new opioid receptor. So all of these types of things are basis for application withdrawal, so keep that in mind. Another basis for application withdrawal is if your application is incomplete. Applications are considered incomplete if they are missing any of the following: go, no-go milestones, a target product profile table, activities for both the UG3 and the UH3 phases of the grants, a budget for each year of the proposed project, including both UG3 and UH3 phases, and then an intellectual property plan attachment. So all of these things that are bulleted must be in your application. And if they're not, then that's a basis for application withdrawal as well. So just a little bit now about administrative review, because you probably are wondering, well, how do we make some of these decisions? So at the end of the UG3 phase, and remember that's the preparatory phase, that's either 1 or 2 years, NIH program staff and leadership will determine if the project should advance to the UH3 phase. NIH program staff and leadership also conduct an annual administrative review. So it's important to think about this, that milestones are assessed and evaluated each year of your awarded project. The reviews are based on successful achievement of milestones, overall feasibility of project management, including biomarker work, and considering data that may not have been captured in the milestones. Reviews are also based on the competitive landscape for the disease indication and the drug target, program priorities, and availability of funds. Milestones, as we mentioned, must be included in the grant application. They are used for measuring success in achieving each of the research plan's key objectives. 1 or more milestones should be used for each key objective. For each milestone, it's important to provide details on the methods, assumptions, experimental designs, and data analysis plans that are the basis for the milestone. It's also important to specify the quantitative criteria for measuring success and the related rationale for those quantitative criteria. Those criteria should be robust and they should be consistent with the state of the art in the field. The quantitative criteria for success in the milestones will be used for making these go, no-go decisions, and this should be specified. It's also when you're talking about your milestones and proposing your milestones in your application for the UG3 to UH3 transition, those especially should be stated that they're go, no-go for the transition. And of course, they must contain quantitative criteria that you've thought through that are realistic, and still make sense and are rigorous. Milestones should be an important component of each administrative decision to transition to UH3. And I should say they are an important component for that. And another thing that's important is to specify the timeline for each milestone. Sometimes people provide a timeline for the entire project and they embed the milestones in the timeline. That can be very helpful for reviewers. And there should be at least one milestone each year. Another thing to point out is milestones will be further refined after award with the NIH staff and the lead development team that's formed around the awarded project. Another important thing to think about is our intellectual property expectations. So inventorship is determined per US patent law. The NIH Pain Therapeutic Development Program has no stake in the intellectual property. We want it all to go to the PI and the PI's institution. So prior to grant award, the PI's institution must have upfront intellectual property agreements in place with all potential inventors. These agreements should address who will hold title to the intellectual property, a new chemical matter or biologics or use, and they should also address royalty arrangements. Intellectual property agreements should aim for unencumbered intellectual property, consistent with our program goal to create a licensable product. So here are a few hints about your application. So please ensure that your application has separate budgets for each year of the grant. For example, a grant of 5 years must have 5 separate sets of budget sheets, plus the cumulative budget. It should be clearly indicated within a table which activities will be conducted by the PI and associated personnel, funded by the UG3 award, in other words, and which activities will be conducted by NIH contract research organizations and or consultants. So you'll remember with one of the first slides I showed you, talked about the lead development team that's formed around the grant and the investigators, institution, and team. So you can choose, as I mentioned, the NIH CROs that you would like to have help you. And you should indicate in the table which major activities you would like to do, you and your team would like to do, and which ones you would like to have the NIH CROs do. And if you indicate that you will use an NIH contract, so that's the other thing to be very clear about. You do not have to use one of our NIH contracts. You could propose to do contracting yourself or do all of it within your team and your subawards. So anything you propose to do yourselves must be in your budget. But if you're proposing to do some of these things through our contracts, then you do not put it in the budget. So another important thing to remember is a table of proposed activities should be included in your grant that provide the following information about your hits or your leads or your lead candidate, activity of your lead or your candidate, looking at the optimization, assays, PK, things like that. You should include things like number of samples per month that you can do with your assays, the source of your leads for your hits, and the source of who is going to actually be doing some of these activities, whether it's going to be your lab, subawardee of NIH contractor, and the advancement criteria. A target product profile table should and must be included in your grant application. You should make sure that you've addressed intellectual property, as we just discussed, in the other attachment section. So that doesn't go in your research strategy section. If you are asking for NIH to conduct a clinical trial under the NIH contracts, then mark application clinical trial no. So you'll see when you look at your grant application form that you can indicate whether you're going to be conducting a clinical trial or not. And so again, If you want NIH to conduct a clinical trial, say no for that particular question. If you're planning to subcontract or conduct your own trial, indicate this will be a delayed onset clinical trial, and then follow the instructions in the form for the information required for that. And it also is important to ensure a data sharing and management plan that complies with HEAL guidelines and NIH data management and sharing policy. And the HEAL guidelines and the notice that talks about the NIH data management sharing policy, the links are provided in this particular slide. And it's also, those links are, I think that all of that's available in your application form as well. Another set of things to do is really, I can't stress enough, read the funding opportunity announcement very carefully. There's a lot of detailed information in there, and there are links to forms and guidances that you need to know. And another important thing, even if you're reading the funding opportunity announcement very carefully, you may still have questions, and most people do. So do not hesitate to discuss your proposal with us and with the program directors. And our last slide gives contact information for them. You should stick to the page limit that's indicated in the instructions on the form. And usually for the research strategy, that's 12 pages. You should also provide a robust description of your scientific premise. So explicitly discuss, and when you're talking about your argument, essentially for why you want to do the proposal and why it's worthwhile to fund the proposal. Also discuss the quality of the data presented in prior publications in a detailed manner. Rigor is another important thing to address. You should detail the controls being used for each type of experiment and appropriately highlight potential confounds like surgery exposure, genotype, culture to culture variability, and human placebo effects. Should include details within the experimental design about the reduction of potential bias, including blinding, randomization, and exclusion criteria, and describe the source of the data on which sample size estimation or power analysis is based, and details about the analysis itself. Additional things to do are to complete the required registrations at least 6 to 8 weeks in advance of receipt dates. Consider submitting your application early. I know you often get busy and you have to do things at the last minute, but things happen, trust me. So it gives you a chance to react to issues that might result in your application being withdrawn if you submit early. Discuss your budget with NIH staff if you have questions, and definitely be talking with your tech transfer business development group because you need to plan for funding patents and licensing activities. Things not to do is do not plan for this to be the sole funding in your lab. Milestone-driven programs, as we mentioned, can end abruptly if milestones are not met. Don't forget to address use of NIH contract resources. Don't plan for NIH contracts to do disease biology. We assume that is what you or your subcontractors or subawards will do. Please do not include NIH contracts in your application budget. Again, if NIH is sponsoring the contracts, that is free. Do not reach out to planned NIH contractors for letters of support. So this is the last slide, and this shows you all of the people that are involved in the pain therapeutics development program and their contact the contact information for the people who are the primary contacts for the grant. So I'm the program lead, and my contact information is here. Chuck Sywin is the director of the Small Molecule Neurotherapeutic Development Program, under which the PTDP program exists and shares infrastructure. And his contact is there. Carolyn Bondar is our health program specialist. Her contact info is there. And Wakanda Medley is our operations coordinator, who is also coordinating this webinar. And then also our scientific program managers we don't have the contact information for them on here, but it's easy to get them through our website. So program managers that work within the PTDP are Mohammed Hachicha, Pascal Lang, Arissa O'Neill-Matherin, Shamsi Race, Rangarajan, Matthew Rice, who's helping us with this particular webinar right now, and Rebecca Ruth. And then we mentioned that there are many NIH institutes involved in this program, and the representatives for those institutes are also listed on this slide. So with that, I'd like to stop and see if you have any questions.
Yeah, oh, sorry, Matt. Real quick before you start on the questions, I just want to remind everyone to submit their questions in the chat section below, and that this presentation will be distributed next week. Thank you.
Thank you, Rhonda. So, Marianne, we have a couple of questions in the chat so far. The first is, can a medicinal chemist submit compounds for specific screening and testing, although they may not have a HEAL grant?
No, that is out of scope for this particular program. If you're interested in screening of compounds as a standalone thing, there is a program through NIMH that is Brian Ross, I think psycho, I can't remember. But anyway, it's a program, it's free to you, and it's I think it's psychiatric screening program or psychiatric asset screening program. I can't remember for sure the name of it, but that would be one option. Or else the other possibility is actually just to contract that out yourself. But we do not, our contracts don't cover that screening either.
The second question is, what's a standard size of FTE team that will be offered for MedChem development if using the NIH contract?
So it varies with the stage of medicinal chemistry that's involved. So we divide the contract into 3 stages. One is exploratory, another is hit to lead, and the third is lead optimization. And the number of chemists increase with each of those stages. So if you're coming in at the exploratory stage where, you have some hint of, you have decent starting material, and it looks like you have a place to go with the hits that you have, but you're pretty much at the beginning still. You probably get 1 to 2 FTEs, and then you would get more than that as you move from hit to lead to lead optimization.
Okay, I actually have a direct question. Supported by federal grants, we have discovered MAB hips in demonstrated their efficacy in animal studies. We wonder whether previous fundings from NIH and DoD present a current concern of overlaps with future HEAL proposal projects.
So it has to be current overlap. If there isn't, to be an issue, it has, because you need to provide your other support. And if that other support is still, is concurrent with this particular funding, then that would be, an issue, and we could resolve it by, just subtracting the funding for the things that are overlap. And we can't fund 2 things at this, 2 similar things at the same time. So hopefully that answered that question.
We've had another question in the chat. I developed a biologic that has been used clinically and currently manufactured GMP for non-pain related clinical use. With colleagues at my academic institute, we filed IP on use of this biologic to treat pain. While we are not making the biologic but have access to it, are we eligible to apply for the biologic identified looking to prove efficacy and biological activity for pain inhibition?
So I think it depends on, this is one of those cases where it's good to call me or set up a Zoom call with me to go over it, because it depends on what you need done. If you want support just to get efficacy, just for your preclinical proof of concept, there are other programs. There's an R61 program that is part of the HEAL initiative as well, that would fund something like that. Because we fund the process. On the other hand, if you want to get efficacy, but you also want to profile the biologic more carefully in terms of safety, distribution, pharmacokinetics, maybe, if it's a gene therapy shedding, things like that. And if you want to take it into IND submission activities, then that would be something we would support. So it just depends on the scope of what you want to do.
All right. Next question is, for the clinical trial phase, how can we get information regarding what the NIH contractors can do?
You would have to talk to us about that because you can't, as we mentioned, you can't directly contact the contractors. So I think that's one of those things where it is really important to talk to us about that, because you should make a strategic decision about what would best serve the needs of your potential proposal.
That's currently all that I see in the chat. Rakonda, do you have anything else that's been sent your direction?
I do not. We can give it a couple more minutes. Okay, we haven't received anything else, Marianne. I think it's safe to say we can end it. Give it some more.
Maybe let's give people maybe like 2-3 more minutes.
Sure. And someone just asked, will we have a recording? Yes, we will. That will be sent out next week.
And Rakonda, the slides will be available on the website as well, right?
Yes. Yep.
Well, Rakonda, I think we could probably end this since it looks like people are leaving now. And again, anyone who's interested and you want to learn more, please don't hesitate to contact us. We really want to facilitate drug discovery to treat pain. So anyway, thanks everyone.
Thank you. Have a good day.